The time has come to dive into adaptive immunity head first. So here we are, innate versus adaptive. On the adaptive side, what we care about here, somehow the adaptive immune system defeats everything. Anything that isn't part of the body can get attacked by the adaptive immune system, and it learns each time. So how does that work? I mean, a big goal of this video and these lectures is to give you the tools to understand how it works, how it does both of those things. This one is a little more simple, it just involves memory cells, but how does activation happen in the first place? And that's a complicated thing. We'll get into it. So the adaptive immune system has lots of different cells. It has to do complicated things. So it has to distinguish between you and the things that are invading you without using receptors. Without using pattern recognition receptors. It has to work without patterns. It has no patterns to look for. And that's the whole point. It's looking for things that aren't showing the normal patterns. So how is that even possible? And again, it has to remember the infections so that it can respond faster the second time. Here's some vocabulary that's going to be important. Look back at what receptors are. A co-receptor is a thing that's going to stabilize a receptor interaction. What is self? Well, self is the concept that some molecules are from us in our cells and other molecules are not. And the immune system needs to be able to tell the difference. So the innate immune system uses patterns to see things like bacteria and viruses. By definition, those are not self. So it won't attack us because we are not bacteria and viruses. The adaptive immune system also needs to avoid attacking us while being able to attack everything else. So it needs to distinguish between self and everything else. A fundamental part of how the adaptive immune system works is antigen presentation. So we're going to look at cells that are going to display antigens to other cells. So what does it mean to display an antigen? That means, it means to show it. What is an antigen? An antigen is something that triggers an immune response. So a bacterial cell is an antigen. A virion is an antigen. A protein toxin is an antigen. What is an epitope? Well, an epitope is the fragment of an antigen that would be detected by a particular antibody or T-cell receptor. So an epitope is a small part of an antigen. An epitope is the individual part that gets detected by a receptor. The antigen is the whole thing that can trigger immune response. I have this in here because most of what we're talking about with innate immunity, most of what we're talking about with adaptive immunity is proteins. The adaptive immune system is very good at identifying proteins. That's really what it's for. It's not as good at identifying carbohydrates. So we will be talking mostly about proteins. So the first part of the adaptive immunity we'll talk about is antibodies. Antibodies are amazing. They're these protein complexes that are really powerful for defending us. They are one of the most powerful single things we have. So all the types of antibodies share this idea that they have regular fragments. So proteins, like this red part here, this protein would be the same from antibody to antibody. So this is not a variable fragment. And then here, where they have receptors for their epitopes, the antigen binding site, so to speak, this is extremely variable. every cell makes a different version of this. So the antibodies that come from one plasma cell have one version of this. The antibodies that come from a different plasma cell might have a completely different version of this. And this is what makes them specific. This is what they use to identify antigens. So, I guess, well, whatever. What do antibodies do? That's a good place to start. There are a lot of things they do, and they do all of it by attaching to pathogens or to whatever. They attach to it. That's their job. So one thing this can accomplish is they can opsonize anything for phagocytosis. So macrophages and neutrophils will be much more likely to attack a bacterium or a fungal cell or whatever if it is opsonized by antibodies. It can also, if you imagine an antibody binding to spike proteins on a virion, that's going to interfere with attachment to cells. That's going to interfere with the viral replication cycle. If we have bacteria in us that can use flagella to move, we can stick two bacteria together using an antibody. So if we go back, each antibody has at least two identical binding sites. So this could attach to one bacterium, this could attach to another. That sticks two bacterial cells together, and that restricts their motility. They can't, like bacteria can't work together to swim in the same direction. So if you stick two of them together, they're going to be making different decisions, and those decisions will restrict them a lot. When it comes to things like protein toxins, like the tetanus toxin, or, well, that's the best example, the tetanus toxin. The tetanus toxin is a protein. It's a protein that does things to our neurons. And if it's coated with antibody proteins, it cannot bind to its normal receptors. So protein toxins can be completely neutralized by antibodies. And lots of them can stick together until they form a big enough network of antibodies and protein toxins that they become insoluble. They precipitate, and this allows phagocytes to phagocytose them. And they can also initiate complement cascades. So antibodies can directly initiate complement cascades. So this diagram shows those things. This would be a virion coated by antibodies, and there's no way these spike proteins are going to be able to interact with the cell. This would be a big protein toxin with antibodies attaching to it, sticking off of it. There's no way this is going to interact with the cells normally either. So this is how we can inactivate the tetanus toxin. This bacterial cell is now opsonized, and it's going to be much tastier to a macrophage or neutrophil. This is something kind of amazing that bacteria do. Antibodies do. They stick bacteria or virions together. And once you have a large clump of these things, they're going to behave differently than they would individually. And this is actually something that clinical diagnostic kits will use. They will have antibodies for a specific part of a pathogen that's been synthesized in a laboratory. And if that pathogen is present, the antibodies will agglutinate the cells. And that'll be immediately visible. The cells will all precipitate. And if that pathogen is not present, the cells will not agglutinate. So that's kind of cool. And then it's also possible to make something insoluble. Make something soluble, like a little peptide toxin or something. If you attach enough antibodies to it, you make this big network, and this whole thing can become insoluble. It can precipitate, and that allows a neutrophil or macrophage to eat it. Okay, so back to major adaptive cells. This is the big list. One thing I'm going to point out is that there are different ways of writing this helper type 1 T cell. I posted these slides. If you're seeing this in 2021, I posted these slides and didn't notice that I hadn't updated this. The better way is capital T lowercase h1. That's how they do it in the literature. This is how they did it in some textbooks I was using 10 years ago. So, good enough. We will get to all of these. We've already looked at dendritic cells. Everything here except the dendritic cell, these are all lymphocytes. And lymphocytes are really unusual in their construction and behavior. And you need to keep an open mind to understand what they do, because it's very counterintuitive. You can build up a whole picture of how the adaptive immune system works, but you really have to start with what a lymphocyte is and what it isn't. So let's look at a lymphocyte. If you look first at this diagram down at the bottom, there are 3 different cells, 3 different lymphocytes have been drawn. And this is how I always start teaching people immunity if I'm in a classroom. I draw 3 lymphocytes, because what this does, it allows me to put their surface receptors on them. So these are B cells, B lymphocytes, and they have what's called a B cell receptor on them. Realistically, each lymphocyte would have roughly 100,000 copies of this B cell receptor on it. And if you notice, this cell has brown, squarish B cell receptors. This cell is drawn with red, circular, or like half-moon receptors. And this one has been drawn with blue-green or green triangular receptors. And what the artist is trying to show is that this one has a set of many copies of one B cell receptor. And it doesn't share that with either of these two. These have their own unique B cell receptor, many copies of it, but they're unique. And that is how lymphocytes are created. When they're doing their initial development, they shuffle their DNA around to generate random new B cell receptors or T cell receptors. Here I'm just showing B cells because they're easier to draw, and that's what the book I got this from uses. So this is where to start. You have to keep this in your mind to understand adaptive immunity. You have to have different lymphocytes with their one receptor on their surfaces in order to follow what they're doing. So B cells have B cell receptors, T cells have T cell receptors. And each one of them could make, like, when one of those cells is in its initial development, shuffling its DNA, making its the program for its B cell or T cell receptor for the very first time, there are a huge number of possible receptors it could make. So this square is one of 10 to the 14th different possible shapes could have been. But the cell has picked one, and that's the only one it's ever going to make. B cells and T cells, or B lymphocytes and T lymphocytes, I use those interchangeably. They're named for where scientists have found them and where they're educated. So T cells go through an intense education process in the thymus. So T for thymus. B cells, or B lymphocytes, were found, I think for the first time, they were found in the bursa, which is a chicken organ that humans don't have. I mean, we have bursae in our joints, but we don't have that specific bursa that a chicken has. So anyway, they're named after that, but it's handy that B cells are educated in us in our bone marrow. That's useful because bone marrow starts with B, just like bursa does. So B cells, we can remember, are educated in the bone marrow. And that's the main thing to remember. So once these cells have gone through education, they're able to respond. They're ready to respond. They have not been activated, but they're ready to become activated. And so they're going to wait until it's time for them to activate. So, if they ever respond to epitopes, they'll be activated. But until then, we would consider them educated because they've gone through education and therefore mature. But we call them naive because they haven't been activated yet. So most B cells and T cells are waiting to see if they should ever be activated. That is, most educated B cells and T cells. And most of them will never activate. So our lymphoid organs are absolutely packed with B cells and T cells that will never do anything for us. But they are ready in case we ever need them. So if they never activate, we would call them naive forever. But we would call them mature because they have completed their education process. So what is the education process? I would like to get into that in the next video. So I'll start by reviewing this and then get into education using T cells as the example. But I think this is enough for this one video.